recording. So number one says that you have a model 1. Model 1 has males that are big B, little b are the males. And we said, I hope you're listening. I hear people already not focused, and then they turn in work that makes no sense. And then you have females that are little b, little b. And you have that in this class. These are beetles. I'm not going to draw them. And they mate according to how the scientists set it up. So that's model 1. And I believe, I'm sorry, they have in each of these containers where they're mating them, there's 2 sets. There's where the males are, they're all this way, I'm sorry. The males continue on this side as well, and females continue where you have males and females that look like this, so that what you're getting is only the heterozygote mating with the homozygote, and you're making all the males heterozygote and all the females homozygotes. Then you have another half in another container, some kind of aquarium, still model 1. You're controlling, again, the males and females. I'm going to try to put them all here. You're going to have, you'll have 6 little b with big B, little b. So you have these mating pairs that look like this, where the little bees are female. Wow, just going to go ahead and, okay. All right, I guess I can't help you. Just have to accept that. Just accept that it is what it is. The males are, do you see how they're opposite? These 2 situations are opposite. Do you all see that? Yes or no, please? So they're opposite. So here the males are heterozygous, females are homozygous. Here the males are homozygous and females are heterozygous. All right. So then they ask you a question, and the reason I'm starting from the beginning is I've seen at least one, and probably, I know of at least 2 or 3 Punnett Squares that were done completely wrong. So we need to make sure that we're doing that correctly. I'm assuming that there's a lot more out there that are completely wrong. Okay? So how many mating pairs are illustrated in Model 1? In Model 1, there's 12 mating pairs, right? You have male, let's count them. This mating pair is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there are 12 mating pairs. The next question is compare the patterns in each of the mating pair in Model 1. So Model 1 up there. Who answered the second question, description? How would you describe them? Go ahead. I'm writing it on the board.
He's writing it on the board.
And then for the second.
So I hear you whispering and giggling. That is what's happening in Model 1. Then the question is, they ask you to take Punnett Squares to determine the possible genotypes of the offsprings from these pairs. So basically, let me summarize it here. That would be big B, cross with little b, where this is mom and this is dad. And then the opposite's true down here. So now let's do the Punnett Square. This is what I saw one person check with me, and thank you for checking with me because it's really important that you check your knowledge. We know that you have problems with dihybrid crosses, right? In general, people can't do them in the room. That's not good. I told you I would set you up with a video explaining them. I posted that video. I kind of expect that you're going to watch it, take notes on it. If I have to make it an assignment, I will when we get back. We'll go over it one more time when we get back. I've given you some practice problems. I kind of expect that you're going to be doing those practice problems along with the pedigree. You've had them all week, right? Yes? Yes. Okay, just making sure because sometimes I get confused. So this is what I saw people do. And maybe they did something different. I don't know. But I'm not sure what happened, but they got all kinds of like things like this, right? Something like that going on. This makes no sense. So you should not be doing this. Punnett squares are not just random combinations of letters. They mean something. So you should be thinking about what they mean. So this is a cross between two individuals with two different genotypes. Yeah, big B, little b, crossed with little b. So that's the genotype of the parents. What's each parent going to give their child? How much of that? Half. Good. Side conversations, joking, and partial participation is not appreciated. So the two, half their DNA, they're each going to give half. So I don't understand why there's like little squares with 4 letters in there. If each parent has 2, what is half? 1. So there's big B, little b should be here. Let me spread them out. Little b here. And little b should be here. And then what you should get is something like this for when the mom, in this case, the mom is crossed with the dad. And of course you would do the other possibility because there's two groups there. That's when the mom is homozygous and the dad is heterozygous. And that leads to pretty much the same thing. All right. That's what that answer should have looked like. So here's where you have to do some self-reflection. If this doesn't look like that, if what you have doesn't look like this, then you need to reflect on why you don't understand that. Perhaps sitting with your eyes closed, your head on the table, eyes closed, and pencil in hand might be part of that problem. Another is perhaps having side conversations and something funny to say every 5 minutes or so. So those might be issues. I'm just suggesting some food for thought. So that would be number 3. Then number 4 asks, now for, of course, when people try to do, if you didn't do the Punnett Square right, you couldn't answer the number 4. Number 4 is asking, what's the frequency, right? I think they say frequency. Or if you had one offspring, they say, predict how many of the first generation, if each pair had one offspring, predict how many of the first generation of the offspring would have the following genotypes. So remember, there's 12 pairs, so we'd expect 12 individuals. What percentage do these Punnett Squares predict? What percentage do these Punnett Squares predict?
50%.
50% what?
It'll be dominant.
So heterozygous dominant, correct. What percent do we expect to be homozygous dominant? 0%. And homozygous recessive. So we'd expect to see 0 big B, big B's. We'd expect to see 6 big B, little B's. And we'd expect to see 6 little B, little B's. Because it's out of 12. That's the expectation for number 4. Writing down the answer and getting it right on this lab is not going to help you on a test. Mark my words. I'm recording. I'm going to post it. I'm not mentioning your name because I don't want to embarrass anyone. When we come back and we take that test and we have a conversation with a parent and they ask me, why is my kid not doing that well in class? In the test, but they do well in class on the assignment. This is going to be the example I'm going to use. I'm going to cut out this 30 seconds, a minute of me trying to convince you that you need to focus and pay attention, and then and all the way up until this particular moment, and I'm going to play that for them so that they know what the answer to that question is. Number 5. How likely is the pairing scenario in Model 1 to take place during the natural course of things in the tank? So if it was nature, how likely would it be for us to get 50-50, and because we're organizing them into these 2 different groups? Would they organize themselves into these 2? No, they wouldn't. As we've discussed repeatedly, they wouldn't do that. In nature, they would just mate with each other, right? So then we wouldn't get a 50-50%. So 0, unlikely, very unlikely. I wouldn't say 0. I don't know the habits of these. I don't know if there's any sexual preference with these beasts. We're going to assume that it's very unlikely. Why is Model 1 labeled selective mating? The reason it's labeled that, as I've already told you the answer to that, is because the scientists are selecting. It's artificial selection. As we saw yesterday, in nature there is sexual selection, but in this experiment, the scientist is forcing the sexes to mate the way they want them to mate. In nature, like we saw with the birds of paradise, did I show you the birds of paradise before you left yesterday? No. Okay. So I did with the other classes. I'm gonna have to. I'll have to wait till another day because we're going through this Perhaps, I don't know. I don't know when we can do it. All right, so let's look at 6. List 2 other pairings that might occur in the population model 1 if the beetles were allowed to mate naturally. So normally we would get, the pairings we're getting here are big B. Let's look at what we're given. So let's look at let's look at 2 things. They're asking for model 1. And the other option is in nature. So the 2 we looked at is big B crossed with little b, and big B crossed with little b, where this one's female, this one's male, this one's male, and this one's female, right? So those are the 2 we looked at. If we do it in nature, what we would expect to see is the following. The 2 added ones that we'd expect to see in nature is big B crossed with big B. And in nature, we'd also expect to see little b crossed with little b. So we'd expect all 4 of those to be the case. Male, female, what have you. So what's the difference? We know in the other one it was 50-50. Let's do a Punnett Square here, where we do big B, little b, and you should be, obviously, there's at least a group of individuals in the room that really need to practice this. So in this cross, the 2 heterozygotes, what you're going to get is 1 out of 4, or let's just use percentages, 25% are going to be big B. Now that's interesting because in the previous model, we saw that big B was 0. Now, we'd expect to see 50% be big B, little b, and we'd expect to see 25% be little b. That's the genotype, where please remember that these 2 are both black beetles. The only white beetles is this one. All right. So they didn't ask you to do this. I just did that to help you understand where they're going with this. So number 7 says, if the population of beetles in model 1 mated naturally, would your predictions of the offspring in question 4 still be valid? No, it wouldn't. We predicted 6 and 6. What we now have to think about is out of 12, what this model predicts, what this random mating would predict, these guys that met and I didn't even do the other option, which was big B with little b. In other words, the heterozygote with the homozygote would give you this. Oh, I'm sorry. I did this one already, didn't I? Yeah, my bad. I already did that one, and then of course you have one more though to do, which is easy, but I'm going to do it anyways to give you practice. So especially if you had it wrong when you did it on your own, you should go ahead and copy this and get practice. So you see here, if you cross 2 white beetles, then you are 100% of versus 25% versus what did we say, 50-50, right? So in this one, if you cross the heterozygotes, you get 50-50. If you cross the heterozygote with 2 heterozygotes you get 25, 50, 25. If you cross a heterozygote with a homozygous recessive, you get 50, 50. If you cross to two homozygote recessives, you get 100%. So 3 different outcomes. So the answer number 7 is absolutely not. So 7 would be no. They would not be the same. The predictions would be wrong if we tried to use just one Punnett square to predict the outcomes. Number 8 asks a different question. Number 8 asks, discuss in your group the limitations of using a Punnett square for a large population. Summarize the key points. I'm going to give you 2 minutes or 3 minutes to discuss that now. So, can we close the door? I know Christian left. He can come back. When he comes back, somebody's going to have to open it. So number 8 says discuss in groups. You had time to discuss. What do you guys think? What are the limitations of using... Wow, we're just going to ignore me. You're just going to have your side conversation. Even after everything I've said, it's just going to... Wow, okay. Okay. So what is it... Let's just go ahead and do white beetles versus black beetles, I guess. So what's the answer that you discussed in small groups? What answers did you have? Let's see. It's 1:09, probably another minute. And I would have given you time to say, tell me, what is it that you thought when you did it? You talked about it in groups. You had a lot to say a minute ago. Nothing to say now? Nothing to say now? You didn't say... Okay.
So we say there'll be too many variations to put in the Punnett square.
Very good. That's excellent. That's right. He did not just say I know.
My fault. I'm sorry.
So, yeah, do you have another answer? Hold on. Let's hear from everyone, please. Go ahead. Absolutely, right? Did you hear these answers are perfect? Yeah, go ahead. That's exactly good. Get the door for Christian. So yeah, when you're doing a Punnett square, Punnett squares are great for one mating pair, right? But when you have not just one mating pair, but thousands of mating pairs, and you're not sure, Punnett squares tell you if I mate this one with this one, then I have the percent chance of getting this, whatever it is, right? And then percent chance of getting that, percent chance of getting this. So Punnett squares help you with a specific mating, but they don't help you with a whole population. Does that make sense? All right, good. So excellent. I'm glad we're rolling here. All right, took a while, but at least we're rolling. Number 9. Number 9 talks about Model 2, and as I said before we started, Model 2 is what happens in nature. Now you have 2 populations. You have males that have both heterozygote and homozygote, and there's a bunch of them. I think there's 12 of those. And then you have females, 12 males, and there's 12 females that have an even distribution of this and this. So now comparing Model 1 and Model 2, how would you say that how is there a difference? I think we've already established that. I don't feel like I need to write it down, but if you guys know, please answer. Yeah.
It's this time heterozygote males and heterozygote females
Are included in it.
Yeah.
And also homozygote with homozygote.
Yeah.
Now we're gonna have a bunch of different crosses, right? Different combinations. Is that what you were gonna say, Christian?
I was gonna say it was like Did he say it's like about the outcomes?
Oh, I don't know. He didn't mention the outcomes. He says now we're going to mate heterozygote with heterozygote, homozygote with homozygote. That was the only thing that was left. Those two were left out of the initial experiment, right? In Model 1.
Y'all just gonna say it's more outcome, more chances of like different outcomes.
That is also correct. So there's a lot more possible outcomes now. For instance, in Model 1, we could not get a homozygous dominant. But now in Model 2, we can't. So that's one difference. All right. So that's a big deal. There's no artificial selection. That's bullet point number 1. Bullet point number 2, it's more like nature. And bullet point number 3, it's more like nature. And bullet point number 3, there are 2 more possible crosses. And bullet point number 4, many more possible outcomes. I think that includes everybody's statement. That includes both Christian and Debarachi's statements of having more individuals that can cross and having a much more complicated outcome from the matings. All right, everybody good with that? All right, so that's number 9. Number 10 says individually match up 12 mating pairs from the population in Model 2 that might occur in a natural random mating situation. Individually match up 12 mating pairs from the population in Model 2 that might occur in a random. So you guys got to do that. I think you can handle doing number 10, number 11. I'll post the answers. I will go through this myself and post the rest of these answers before tomorrow morning. Okay? I will be in esports tonight. They're doing a tournament. So there'll be some noise in the background, but at least you'll have the answers. Okay? All right. So go and work on this for the rest of the period. All right, number 10 says individually match up the 12 mating pairs from the population Model 2 that might occur in a natural random mating situation. So you'd expect to see big B, little b crossing with female big B, little b. Okay, that's fair. Notice what I'm doing is I'm just picking random. I'm just going to go across. It's hard really to do random. Believe it or not, it's actually even computer programs, it's very difficult to create something at random. So I'm just going to go systematically and just go across and try to make sure I get every possible combination, because anything can happen at random. Remember what we say when we said the flipping a quarter, you could get 15 heads in a row. Randomly, it could happen. Unlikely, but okay. So, or 20 heads in a row, or 1,000 heads in a row, or whatever. So sometimes there are patterns, and just because there's a pattern doesn't mean it's not random. It all depends on how when you're talking about randomness, the question is how likely is it to be random? In this case, since we're trying to create something random and we really can't, I'm just going to go across and put them together, male to female. So we go male, this big B little b is going to be crossed with little b, and then a little b is going to be crossed with a little b. And then finally the big little b with big B little b, which I know we already had, but that's all we could, that's the only possible set of combinations, right? And then we do that 3 times, right, for each row. Male, female, male, female, male, female, etc. So I hope you see that what's going to happen is that as your oh man, what's going to happen is you're going to end up with 3 copies of this particular 3 copies of these combinations, right? So because the rows are going to be about the same. So this is what you'd expect with 12 mating pairs. Again, what's the question? The 12 mating pairs are going to be 1, 2, 3, 4. There's going to be 4 really different types of mating, or actually only 3 different types of mating pairs, but there you go. All right. So that is the individual mating pairs. Now compare your set of mating pairs with other members of your group. Did your mating scheme match anyone else's in the group? I think what's going to happen is that if you mated them, these were the only possibilities. You might have this one being male, this one being female, then there might be a different combination male, females, but in general, these are going to be the only ways really they're going to be able to mix, because there's a limited number of ways you're going to be able to mix these 3 different types, these 3 different genotypes: big B, and little b, little b. Actually, there's only 2, right, types of mating types. But so the question is, do you put big B, little b with big B, little b? Do you put big B, little b with little b, little b? Do you put little b with little b, little b? You know, those are pretty much the only ways you're going to be able to mix these 3. And the rest are just going to be repeats of one another. Now, is the heterozygote male or is the heterozygote female? Is the heterozygote, is it homozygous dominant female, homozygous dominant male? You know, which one's which? I think it all works out. You could actually label all these and you'd eventually get all the same 12. So when we're looking at this set of problems, you know, at this set of questions, we'd have to consider the population's changing. Now we're not limited to these 12, right? So let's go ahead and read that. So it says, when it comes to mating in natural populations with hundreds or even millions of individuals, it is difficult, maybe even impossible, to think of all mating scenarios. After several generations of leaving things up to nature, the alleles that are present in the population will become more and more randomized. Statistics can help biologists predict the outcome of populations when this randomization has occurred. If the population is particularly non-random, to start, this randomization may take several generations. All right, what do they mean by particularly non-random, right? And what it means to be particularly not random is that perhaps you had a bottleneck event, right? Or perhaps you had a founder. So you have this island, and you know, you have palm trees, coconuts, and you have birds come in, they're flying in from different places, and maybe you get a lizard or 2 on the island. So that's the beginning of the population. Obviously, the island's has to be much bigger in order to support any bigger population, but let's assume that this is the beginning. You have these 2 male, female, like Noah's Ark, 2 of every creature, right? 2 birds, 2 turtles, 2 whatever. So that being said, the question then becomes, what if this is going to be the beginning population, is that as that population grows, how are the alleles going to separate? How do we randomize those alleles? Well, it's going to take many generations, many generations of intermixing, of sex occurring, and mixing up the genes and crossing over, and all the things we had talked about up to this point. Many generations of that happening in order to get the alleles distributed around the whole population. So usually whenever you have a new group coming in, let's say a group immigrates or emigrates into a new region, and so you have this area, and you used to have pretty random distribution of individuals in the population, pretty random. Again, is it anything completely random? It's so hard to make something random. I can tell you it's just really hard. But what happens if you have a group that comes in and they're clustered, individuals cluster together, right? People like what they know, and this is true probably of animals and likely like plants. If something somehow gets transported, over time, these genes, these individuals with these purple alleles, will mix. They will distribute. They will move in different directions within the available space. And as they have children, and those children move, and individuals or families move, after some time, you'll get that randomization again. That's what they mean. And the more non-random they are, so the more clustered they are, those alleles are, or the more, in the case of a founding event on a group of islands, or in the case of some group of individuals coming into a larger population, those alleles are going to move through the population over time and spread out and become randomized. So it's going to be hard to follow them, to follow whether those what that distribution of alleles is, and whether evolution is actually occurring or not. So how many alleles are in the population in model 2? So the number of alleles, how many alleles do we have available in this population of males and females in this random assortment? So we got 1, 2, 3, 4, 5, 6. So there's 2 per individual, right? So there's 4 by 3, so there's 12 individuals here, and there's 12 individuals here, 12 males and 12 females. So we have a total of 24 individuals, right? So 24 individuals times 2 alleles each, right? So each one has 2 alleles. So we have a total of 48 alleles available to us, either big B's or little b's. I didn't count how many big B's or little b's there are, but that's what we have. What is the probability of an offspring in Model 2 population getting a dominant allele? So what's the probability of some of these the interactions of these individuals, these matings, of getting a dominant allele? So let's think about that. So if you have 48 alleles total, how many dominant alleles are there to give? So let's count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there's 12 dominant alleles, 12 B's, right? So what's the probability that these individuals are going to have babies and produce, this population of males and females is going to have babies that have the dominant allele? So 12 I just counted 12. I believe I was right on that. Let me double check. Sorry, my wife's cooking dinner. The dogs are moving around. But 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. That's right. So there's 12 alleles, 12 capital B's, 12 possible dominant alleles that could be given. And if you count the recessive ones, by the way, just as an aside, they don't ask that in the question, but 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Oh, sorry, I miscounted. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36. So 36 little b's. And that makes sense since the total was supposed to be 48, and I could have just subtracted 12 from 48 and got 36, but I wanted you to see that you're dealing with 36 alleles, lowercase alleles. So what are the odds that any child that all these parents have are going to have a big B at all, and therefore be black, by the way, because it is the dominant allele? All right, well, the odds are 12 out of 48. And that equals 1. Well, that's going to be 0.25 or equal to 1 divided by 4, which is equal to 0.25 or 25% chance of having for these individuals to be able to have a child with a dominant allele. What's the probability of the offspring in Model 2 of getting a recessive gene? Well, luckily, I already did the math for that. It's going to be 36 out of 48, which is equal to 3 out of 4, or 0.75%. Because remember, there's only dominant or recessive, so together they have to equal 100%, right? So together they have to equal 100%. And so if you have 25% dominant, you have to have 75% recessive. I hope that makes sense. If not, see me in class, see me tomorrow during advisory, during tutoring. So you got 0.25 and 0.75. If P is used to represent the frequency of the dominant allele, and Q is used to represent the frequency of the recessive allele, then what will P plus Q equal? Well, we just said it. It makes common sense, but if you have however many little b's you have and how many little big B's you have, out of the total, they have to equal 1 or 100%, right? Because when you 3 out of 4 plus 1 out of 4 is 1. 0.75 plus 0.25 is 1. If you did percentages, which would be 0.25 times 100%, that would equal 25%. And 0.75 plus or times 100% would equal 75%. Together, they have to equal 100%. It's just a common sense idea that if you have only big B's or little B's to give, there's only 2 alleles, you have a population, everyone has 2, you have to have the number of B's, the frequency of B's plus the frequency of little big B's plus the frequency of little b must be equal to 1, because that would mean that you have 100% of all the alleles, and of course you do if you're accounting for their frequency in the population. So out of 48, the total has to equal 48. If P is used to represent the frequency of the dominant allele, Q will equal 1, as I said. P plus Q must equal 1. Use your knowledge of statistics to calculate the probability of an offspring in Model 2 of having each of these genotypes. Support your answer with mathematical equations. Don't forget, there are two ways of getting heterozygous offspring, big B, little b, or little b, big B. Again, that was the whole point of the first part of this exercise, for you to realize that you can get a big B from mom and a little b from dad, or you can get a little b from mom and a big B from dad. So you can get them two different ways. So remembering your Punnett Square, you get 0.25% of the total is going to be big B for head. Well, hold on a second. Let me erase that. You have to remember that when you're talking about this population and you have this combination of genes going on and it's random, you'd expect to see that the numbers are going to equal 1 or 100%. So let's think about it. If little b is P, big B, little b is, there's two ways of doing it, which is why they gave you that clue, is 2P times Q, right? And little b is Q squared. Oh yeah, that's P squared, by the way. So what you end up getting is 2PQ, 2 times P times Q. Let me give you some more distance here. 2 times P times Q. So P squared is equal to 0.625. And when you take a look at these and you add them all together, right? So again, just to remind you what we're looking at here. This one is the heterozygote. This one is the dominant, the chances of getting the dominant, two dominants in a row. There we go. And then this Q squared is 0.5625. And I can add them together, and I get the number 1. All right. So that's why they say here to check your answer, and by the way, this answer would go here, right? To check your answer, they say your sum should be equal to 1. Explain why the sum of your three answers in 16 should be equal to 1. I think we've already said that. If you only have P's and Q's and you have 48 of them, each individual has 2 of them, then their sum must be equal to 100%, which is all this that we discussed up here. So again, the frequency of the components equals 1. The component parts must equal the whole, right? Otherwise, you're missing something. So using P and Q as variables, write the formula for calculating the probability of an offspring from a population having each of the following phenotypes, which I already did already. So it's P squared, it's 2PQ, and I already gave it to you in class, right? Squared. Complete the equation, and that equals 1. That's it. That's the equation I gave you in class, and I told you would never have to memorize it because I give it to you in class, but there it is. So the next question asks, the equations you just developed, P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1, were first developed by G.H. Hardy and William Weinberg. They represent the distribution of alleles in a population when the population is large, right? When there's mating is random. Remember the 5 postulates. This is when this Hardy-Weinberg is true, when there is no evolution, right? All genotypes are equally likely to reproduce. No organisms enter or leave the population. No mutations occur. In other words, the group of alleles available in a population must be very stable from generation to generation. if the distribution of genotypes in a population matches the predicted Hardy-Weinberg equation, then the population is said to be in Hardy-Weinberg equilibrium. Now, this is really key. When a population, any random population, is in Hardy-Weinberg equilibrium, and by the way, in nature, it's not going to be often, and when it is, it's likely to change before too long. Time is relative, right? It takes at least a generation. With humans, that would mean, 18 years? Let's just say, and I know it's less than that, but let's say, having a baby, that baby getting old enough to have babies, right? At least a generation for the alleles to change frequency, unless some drastic thing happens like genetic drift, right? Genetic drift could wipe out a whole section of a population in a swoop, but it would have to be a really big event if it's a really large population. So if the distribution of genotypes in a population does not match the predicted Hardy-Weinberg equation, then the population is said to be evolving. So it's when this is not true. It's then when this is, So when this is not true, then evolution is occurring. Now, consider the requirements for a population to be in Hardy-Weinberg equilibrium. In the natural world, are populations likely to be in Hardy-Weinberg equilibrium? Justify your reasoning. As we said before, no, it's not, because at least one of these things is always occurring. You're not gonna have random mating. There are choices being made. We watched the Birds of Paradise, most of us, and we saw them mate and make choices. You know, populations tend to be large. Especially with humans, you're not going to have a small population. If you do, then maybe. No mutations occurring. Mutations always occur. No organisms enter or leave the population. No immigration. No emigration. Highly unlikely, right? You're going to, in any kind of population that we're used to, you're going to see individuals moving in and leaving. So let's go ahead and take a look at sickle cell anemia, genetic disease. Sickle cell allele is recessive, but individuals with homozygous recessive often die prematurely up to the disease. This is in the past. It often is relative. So I'm sure a lot of us have friends or families that have sickle cell. It's not necessarily deadly. It's certainly an issue, but let's put things in perspective here. But if individuals with the homozygous recessive genotype, little s, often die, those people are said to be homozygous recessive, right? And affects the population, about 9% of the population in Africa. So about 9% of the population in Africa, and they tend to be around places where there's a lot of mosquitoes, by the way. 9% of the population in Africa have this little s. That means, think about what that means. I'm just going to pause here. Little s. What is little s? Pause. Okay, that's the homozygous recessive. So we're talking about what's 9%? What is the number 9% mean? Well, it's that 9% in this case is the probability of homozygous recessive, of getting little s in the case. So what we're seeing is 9%. What we see is Q squared. So Q squared is equal to 0.09. Where did I get 0.09? 9% is 0.09 times 100%, right? That's how you get 9%. So it's 9 out of 100. And we move the decimal twice, because if you divide 9 by 100, you do move the decimal twice to the left, you'd get 0.09. So the frequency of the recessive allele in the population, so just Q by itself would be what? Let's pause for a second. So how would I get Q by itself? I'd have to take the square root of both sides. So when we take the square root of a square, we get the variable by itself. So square root of Q squared is Q. The square root of 0.09 is 0.03. You can do the math on your calculator. So Q is equal to 0.03. What is the frequency of the dominant allele, P? Well, for that, you have to use the other equation. There's two equations you got up here. By the way, you'll get both of them on the test. P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. So if P, let me change the white here. If P plus Q equals 1, and Q is 0.03, what's P? Well, it's 1 minus 0.03. And that's P must be equal to 0.097. I'm sorry, that's 0.97, not 0.97. How did I get that? Just to remind you, to get P by itself, I subtracted both 0.03 from both sides. 0.03 minus 0.03 is 0. 1 minus 0.03 is 0.97, and that's P. So this is our P. Is point. So we have two values here. We have 0.97 and 0.03. So this is the frequency of the recessive allele, and the other is the frequency of the dominant allele, right? So what's the frequency? So this would be here, this would be equal to 0.97. What's the frequency of the homozygous dominant individual in African population? Homozygous dominant would be P squared, which would equal 0.97 squared. So that would be 0.9409. And there really should be a 0 in front of all this, but I'm kind of doing this. Did I get that right? Okay, that would be 94. I don't know why I changed that to 7, but there it is. So this is the frequency of the homozygous dominant big S, right? Remember, I squared it because there's two. That's the probability of each individual event happening concurrently. So whenever you have this and this versus this or this, right? So you're multiplying. Remember the sum and the multiplication rules of statistics. But let's move on. The frequency of the homozygous dominant allele in the African population is equal to 0.94. I'm just going to round it off to 0.94 or 94%. What is the frequency of the heterozygous individuals in the African population? Well, again, remembering that Q squared, Q squared is the homozygous recessive. It's 2 times P times Q. That is your heterozygote, or big S, little s. Big S, little s frequency is equal to 2 times P times Q. So what you have is you have this frequency of the heterozygote allele being 0.0582. And then, based on this analysis, is the population in Hardy-Weinberg equilibrium justify your answer? Well, in order to find out if it's in Hardy-Weinberg equilibrium, remember it has to be P squared times or plus 2PQ plus Q squared has to equal 1. We know p squared is, so we know p squared is, let me highlight it here. We know p squared, oops, do a different color. P squared is 0.9409. We actually wrote that here. That's a p squared. So p squared is 0.9409. If you add that to whatever 2pq is, which is here, that's the heterozygote, that would be this number here, which is 0.0582. And that one has to be added to q squared, which was the original number we were given, which is 0.09. Remember, q squared was given to us. That was the probability of having sickle cell. How many people have sickle cell? The only people that had sickle cell disease were q squared people. Remember that we're always looking for the homozygous recessive, because homozygous recessive is easy to identify, and so we can count them easily. So q squared is 0.09, and that was the little s, right, that we talked about, the recessive allele. That percent was 9%, or 0.09. So that would be 0.09. So in order for this to be at equilibrium, in other words, no evolution is occurring, all three of these have to equal 1. Well, let's see if that's true. I don't know if that's true. So what do they equal? Well, it turns out that that number is not 1. It's 1.0891. Now that's daggone close to, that's pretty close to 1. Now the question is this number close enough to 1 that any difference that we see from 1, this 0.0891, this pretty small number, is this significant or not? If it's significant, if it's significantly different from 1, then there's evolution going on. If not, then anything we're measuring is just measurement error, or it's some kind of random cluster or something like that. So how do we tell that? How do we tell if this is a significant difference from that equilibrium from 1? We have to use chi-square. All right, so when we take a look at this, we look at individuals with a heterozygous genotype, big S little for sickle cell, or heterozygote for sickle cell. They have resistance to malaria. It's a serious disease, and it's spread by mosquitoes. So it turns out that in places where there's mosquitoes, you get a lot more sickle cell disease. But the disease, the sickle cell trait, that's this here, having the trait, having the little s, it's actually having, people say having the trait. It's not really having the trait. It's having the gene, the allele, right? People say, do you have the trait? It's actually, do you have the allele? Having the little s gives you resistance to malaria. Malaria kills many, many, many people every year. It's a very harsh little parasite. And not just people, but animals as well. So you have to discuss in your group how this might affect the frequency of the allele in the African population. Summarize your conclusions here. Well, what you would expect in Africa is anywhere there's desert, there not to be mosquitoes. So mosquitoes, as you know, need water, relatively warm temperature, water so they can reproduce. And I don't know if you probably didn't know, they need a specific snail and they need humans as part of their life cycle. They have different stages. But the point is they need water. And so anywhere in Africa where you don't have a lot of standing water, and not just water, but standing water, right? Water that's not moving too much, not like a river, but we're talking like a pond or water in a tire or something like that. The mosquitoes can propagate, they can lay their larvae, and they can multiply. So standing water is important. So it turns out I would expect wherever there's standing water, there's going to be a lot of malaria. And if you're going to have a lot of malaria, you're going to want the heterozygote. You're going to want people that have this trait, because people with big S, malaria gets them. People with little s, well, they get caught by sickle cell. And today, it's especially true if you don't have if you don't have health care, right? So in a lot of the world, hospitals aren't on every corner the way we have them here in Cleveland. So we're very lucky here. But in some places, it's hard to get help. So babies are born and they are sick, and people don't know why, and they might get to the doctor too late or what have you. And so these sickle cell, the result of sickle cell disease can be really harmful. And treatment it's a chronic disease, so you have to manage it. Where sickle cell, the heterozygote for sickle cell is resistant to malaria and doesn't have sickle cell. So this is a sweet spot right here. This is where you want to be if you live in a place that has a lot of these mosquitoes, a specific kind of mosquito that carries malaria. It's that sweet spot in between the two. Well, how do you get the heterozygotes? Remember, you have to, if the heterozygotes are favored, you're going to have a lot of heterozygotes. Let's go ahead and do a simple Punnett Square of a heterozygotes just for argument's sake. And what you'll see is that, just like we know already, you got a good portion, whenever you have two heterozygotes mating, you have a good portion there in the sweet spot, right? These guys. So 50% are going to survive. They're not going to have sickle cell disease, and they're going to be resistant to malaria. So you're going to get 50% of that population surviving. But of course, that means every time they have babies, there's a quarter chance that you're going to get people with sickle cell disease, or people with, or people that might die because of malaria. So every generation, you're going to have these individuals there. In fact, because the heterozygote is what's favored, you end up getting these other gene frequencies in play. All right, so that's the summary of what I would have expected you guys to come up with. We had discussed it in class, so I would have expected you to say something like that. How might this trait affect the values calculated in question 21 and the population and the tendency of the population towards equilibrium? Well, you'd expect that there would be less in places where there's malaria, because malaria is not all over Africa, and the numbers they took, they said 9% of the people in Africa have sickle cell disease. You'd expect that most of them, about 50% of them, would have the trait, and another percentage would have neither the trait, or rather, I hate using that word trait, neither have the allele. In other words, they'd be homozygous dominant. In places where malaria is found, you'd expect that what you'd find is more of these guys, the homozygous dominant, maybe the heterozygotes. You're less and less, you'd have more big S's. So P squared would be bigger and Q squared would be smaller. In places where you have malaria, you'd expect to see the 2PQ to be bigger and Q squared to be smaller. And so the equilibrium point might be different, actually, in different places, but you might actually see evolution occurring depending if malaria is coming or is it going. Have they been able to address malaria, or they've gotten rid of it? So it's really a complicated question, and there's a lot of pieces and parts, so you can have a really intelligent and vibrant discussion and even a debate on what you think might be happening. All right, so let's stop here and consider a beetle population model 2. Imagine a change occurred in the beetle ecosystem that made it easier for predators to spot white beetles, and 6 of the white beetles were lost. Now let's talk about that population 2 again. If we look at population 2, we lost. If we lost 6 of them, remember we said there was how many? 1, 2, 3, 4, 5, 6, 7, 8. So there's a total of 24. Just to remind ourselves what we were talking about earlier, 24 total individuals. So each of them have 2 alleles. So there's a total of 48 alleles. Now, if we got rid, if 6 white beetles died, now you're down to, so you got 18, because you don't have that anymore. It's minus 24 minus 6. Now you only have 18 individuals. And out of those 18, you double that, and there's 36 total alleles. Now, so we've reduced the population, we've reduced the number of alleles. This is no longer true, right? We reduced the population, reduced the number of alleles, but we reduced them not evenly. We didn't reduce. Notice we got rid of only 6 white ones. That means we got rid of a lot of little bees in the population. How many little bees? So let's talk about how many we have left, right? So we have 1, 2, 3, 4, 5, 6. 6 times 2 is 12. So we have at least 12 little bees from just the white ones. And then we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And then we have 12. We have 6 more little bees. 12 minus 12 divided by 2, because only half of their genes are little b. That's 18. So there's 18 little bees now. Little bees are 18 out of 36. All right. So that means big B has to be 18 out of 36. So now we have 50-50. Now there's a 50% 50% little b and 50% big B by getting rid of those 6 genes. So those 6 individuals with homozygous recessive genes. Before, it was 75%. Remember, initially we said it was 75% in the population of little bees, and there were 25% big bees. So just by getting rid of those 6 natural selection. This is natural selection. The birds are able to pick out the white beetles more often because of the environmental change. What happens is that you went from 75% little bee to 50% little bee. You went from 25% big bee to 50% big bee. So there's a really big change here. So the next question is let's consider what they say. Predict what the genotype frequency in the population after this event. Well, we just talked about it. We'd have the genotype frequency. That's another little bit of a story. So let's go ahead and do that. So we know that the little b allele frequency is 50% or 0.5. We know big B is equal to 0.5. Well, little b squared is kind of easy to figure out as well. If we go back to model 2, how many little b squares do you have? I have 1, 2, 3, 4, 5, 6, right? 6 out of 18. So we know that little b is found in 0.5 and a frequency of 0.5, 50%, and 50% big B. So what would be the predicted genotype frequency in a population after this event? Well, we would expect that after this event occurred, we would have 0.5, so 50% squared is 0.25. So we'd have 0.25. That would be Q squared. P squared is going to be the same because they're both 50%. It's 0.25. So that means that 2PQ would equal 0.75, right? Oh, no, I'm sorry, 0.5. I don't know what I'm thinking. Because they have to equal 1, right? So if I know it's 25% Q and 25% recessive, homozygous recessive, and it's 25% homozygous dominant, then the heterozygote has to be the other half because they have to equal 1. So what would I predict? I would predict point P squared equal 0.25, Q squared equals 0.25, and 2PQ equals 0.5. Compare your answers in question 22 with those in question 16. How do the answers support your conclusion that this is not in Hardy-Weinberg? So when we look at what happens when natural selection, if natural selection is occurring, evolution is occurring, right? And this is this is the change that happened when evolution occurred. We went from a distribution or an allele frequency that looks something like this, right? That we talked about on an earlier slide, which is about heterozygote. So it would be big B, little b. We expect it to be about, and put equal, about 38%. We'd expect the big B to be equal to 6%, and we'd expected the Q squared to be equal to about, or rather the little b to equal about 56%, give or take some percentages. This is kind of what we expected before. Now evolution happened in this example, in this model. We had evolution happen, and all of a sudden, and after just 6 white beetles die because of this natural selection impact because the environment changed, right? So the ground got darker, and now white beetles can easily be seen. You had this distribution previously. Now you get rid of the white beetles. They start to get chosen. They become less favorable to survive. And all of a sudden, Q squared, or little b, is not 56%. It's only 25% of the population. Well, that makes sense. But if that goes down, remember, the others have to go up. The others have to go up in number. If that one goes down in number, the others have to go up in number, right? Or at least the P squared has to go up in number. You have to compensate, right? You can't just bring Q squared, which is little b frequency, up to 56%, from 56 to 25, and then leave this at 6 and 38. You'd have to bring the heterozygote up from 38 to 50, and the P squared from 6 to 25%. These had to come up in order to compensate for this one coming down. So the white beetles now became less in the population. You lost alleles. You lost, and of course in the future, once it stabilizes, you're going to have less of the white beetles, less of the little bees. And if this kept going, if that pressure kept going, then this percent would just keep getting smaller and smaller as all the white beetles were picked out first. And you got less and less little b's until it became background noise. So that's how alleles can come and go in a population. That's how they can increase and decrease in a population. All depends on the pressures of the environment. And that answers this question here. That also explains why we think if we accepted that difference between 1, it should equal 1 and 1.08 that we saw with sickle cell, there's a difference there. If we accept that difference is significant, then it's telling us something is going on that's not within the predicted realm. There's something that it needs to equal 1, so we have more than what we need. So it's likely one of those values, the homozygous recessive, homozygous dominant, or the heterozygote is really not is either not favorable to survive or it's more favorable to survive, and therefore is in larger numbers than it would be if it were just randomly distributed. Now let's look at PTC. PTC is a chemical that you find in that you can find on a slip of paper that you can buy in a science store, and it's also found in certain vegetables, and it tastes really bitter. People really, I believe it's broccoli, but it might be another vegetable, I can't remember. And if you taste it, some people taste it, others don't. You taste it because of a dominant allele. If you have the dominant allele, you taste it. I used to have PTC paper in class, but I don't have any anymore. Big T, if you sampled 215 individuals and determined that 150, you had 215 total, and taster, 215 total, and you determined that 150 were big T, so 150 were big T. We don't know what the other one is, right? We don't know if that's homozygous, dominant, or is that heterozygous? I don't know. But 150 of them were tasters. And 65 could not taste it. Now, these are the more interesting ones, right? These guys, these are they. This is the key. It's that recessive being 65. Because that you do know the genotype for. You know that's little t. That's the homozygous recessive. So the frequency of little t, right, getting 2 little t's in a row is 65 divided by 150. So Q squared is equal to 65 divided by 150. So that's 0.433. So Q squared is 0.433. Where'd I get that? I took the 150, which is, oh no, I did that wrong. Hold on. So this 215 was the total, not 150. So that was my bad. So 65 divided by 215 equals 0.3023, and that's your frequency of little t, or Q squared. So your frequency of Q is the square root of that, right? In order to find the frequency, so they could ask you, what is the frequency of the recessive? They're asking you what the frequency of the recessive allele is, right? So now I got to take the square root of both sides, because in order to find the frequency of the recessive allele, I have to take the square root of both sides, right? And that gives me Q. Square root of Q squared is equal to Q. So the square root of 0.023 is 0.55, which, by the way, would be about 55%. So Q is about 55%. It's about, I'm rounding here. It's about 55%. And so P must be about, that's what that wiggly equal sign equal sign means, by the way, is about. Then therefore the frequency of the dominant allele has to be about 45% or 0.45 because they have to equal 1, right? That's one of the formulas you had. We said I'm gonna make sure you know you're given on the test P plus Q equals 1. So P being the frequency of the dominant and Q being the frequency of the recessive alleles. So then what about the frequency of heterozygous individuals? Well, that, again, as we discussed before, that's going to be 2PQ. And so 2 times P times Q is 0.45 times 0.55 times 2. So that would be about 0.495. I know some of you are sitting there thinking, well, wait a minute, that's 49%. You said 55% is Q, 45% is P. Well, how can you have 49% or 49.5 or 0.495? How can you have 0.495 if the two have to equal 1? But please note that it's P plus Q equals 1, right? That's the single, that's the allele, that's the frequency of the dominant allele. This is the recessive allele. And where this is not the allele, this is the heterozygote. This is the big T, little t. Right? Big T, little t. How many of these do I have? Well, it's about 49.25. You can figure out all kinds of numbers based on this now. Now you can go back and say, well, how many of the 150 are heterozygote? Well, it'd be 0.495 of the total population. So it'd be around 106 individuals out of 215. 150 are big T, so then if you subtract 106 from 150, that would give you how many are big T. Or you could just plug in your equation. You could say what's Q squared, which would be 0.55 squared, and that'll give you the dominant or the recessive, which would be the non-tasters, which should equal 65, right? So this is a way for you to check. That would be a way for you to check your answer to see if you're right. And then here it would be 0.45 squared. So 0.55 squared should give me a percent of 0.55 squared. So let's go ahead and do that just to show you how you can check your answer. If we're right, if all my calculations are correct, and I hope I'm not going to embarrass myself, but we'll see. 0.55 squared, that should give me the recessive alleles. The frequency of recessive alleles. And I should be able to multiply that times 215 and get 65 or something close, because I rounded. There might be some rounding error. So, but 0.55 squared is 0.3025. So already I'm feeling confident because look at what we took a square root of, right? So we got close to the right answer, to the beginning answer. Where do we get off? Why is it 0.3025 now? Because I rounded earlier in the calculations, and so that made a change here at the end. And then if I take 0.3025 and I multiply it times 215, I should get somewhere near 65. And what I get is 65.03. So daggone it, I'm pretty sure I got it well. I did a good job. I came close. So that's how you can check your answer. You're going down one direction, you're making sure you're doing everything right, you make sure you use the right formulas, you're using the P's and the Q's correctly, you're punching the right numbers in. Sometimes you know what you're doing, you just punch the wrong numbers. I've done this several times while I was doing this. Instead of putting plugging in 0.55, you might plug in 0.35 or something like that, which is going to give you a different number. So by going backwards, you can check your answer. Finally, the last question of the day says 60 flowering plants are planted in a flower bed. 40 of the plants are red flowering homozygous dominant. So homozygous. So let's go through, write down the numbers. We got 60 flowering plants. That's the total. You got to remember that because I made that mistake earlier in these calculations. I used the wrong number. 40 are red flowering homozygous dominant. So big R, big R. 20 are white homozygous recessive. So little r equals 20. The plants naturally pollinate and reseed themselves for several years. In a subsequent year, you have 178 red flowering plants. So red flowering. 178 red flowering plants. And what was the other one? And 190 pink flowering plants. And 52 white flowered plants. Use chi-square analysis to determine if the population is hardy Weinberg. Now, here is an example of incomplete dominance. I know I told you that we're going to stick to dominant recessive traits, and we don't want to deal with these other types of inheritance. But it turns out that when you're dealing with incomplete dominance or codominant, and you still only have two alleles, you can still do this. You can still make it work. Because really what you're worried about is the alleles. And what the great thing is, I told you before about codominance, right? The great thing about codominance is that what you see is what you get. This is the great thing about incomplete dominance as well. If you see red, then you know it's big R, big R. It's I'm gonna have to call a mistake on this. I think they made a mistake on this. I don't like it. I'm gonna say don't do this one because there's too many mistakes in this particular question. The problem is they're using the word recessive, and it's not homozygous recessive. This is an incomplete dominant situation because you're getting red. You're getting three phenotypes with two alleles. So this is incomplete dominance. It's not codominance because you're not getting red and white. You're getting red, white, or pink. And I could make this work. I could solve it. I could correct their mistake. Instead of calling it recessive, I can come back and change this to instead of big R and little r to big W, and then pink is RW. I could do that, and then this would be the heterozygote, and this would be the homozygous red, and this homozygous white. And then this would be, you could just pick one. This is Q, this is P, or vice versa, because it doesn't really matter as long as you keep them constant. And this is 2PQ. Because the statistics, the math still works. But I don't like it when they make mistakes like this and they end up confusing the situation, and it takes a long time to resolve it, and people end up not being happy. So let's go ahead and just clog this one out of the discussion, and let's not bring that one up again. Only because it would be fair for them to ask it if they expected you to know, that you could do this, that, this would be PQ squared, this is Q squared, and this is 2PQ. And you know that they have to equal 1, and therefore, are they in Hardy-Weinberg equilibrium? Well, what's the frequency of each of these? All three of those percentages have to equal 1. If they don't equal 1, then they're not in Hardy-Weinberg. And then we'd have to do a chi-square analysis, look at the predicted, look at the actual, compare the actual to the predicted, and look it up and see how many degrees of freedom, and find out what at that point, find out what the critical value is and compare our chi-square value to the critical value. It's just too much. Let's just leave it there. And I think if you understood how to use Hardy-Weinberg, if you're in the AP class, understanding this one, we can go over it in class because it is a valid question. But the non-AP class, I don't want you even to worry about this. I think what's important for you to get out of all this experience is understand this idea that the number of recessive allele, the frequency of recessive alleles, and the frequency of the dominant recessive alleles and the dominant allele have to equal 1, or they have to equal 100%, and that when we're talking about the genotype, the heterozygote being 2PQ, the homozygous dominant being P squared, and the homozygous recessive being Q squared, that all three of these have to equal 1, right? That Q squared plus P squared, P squared plus Q squared plus 2PQ must equal 1. Again, don't panic because these three don't equal 100%. These equal more than 100% because this is not Q squared, right? Well, let's go ahead and do that because that could be a little confusing when you're looking at it. It's another way of checking your answer, right? So when you look at it, at first you're like, wait a minute, those don't equal 1. But that's silly because we're not talking about values there. They're pumped into a formula. So if you took 0.55 and you squared it, that would equal 0.09. Oops. Right? That's what 0.55 squared is. And if you took 0.45 and you squared that, is 0.2025. No, that doesn't seem. 0.55 squared is, that's wrong. Sorry about that. Is 0.3025. I'm doing the other one again just to make sure. 0.45 squared is, that's right. It's 20%, that's 30%, that's 49%. So now add those up, 0.2025 plus 0.3025 plus 0.495 equals 1. So these three numbers, the actual numbers after you've done the squaring, 2 squared plus Q squared, or P squared plus Q squared plus 2PQ do in fact equal 1. So that worked out. All right. So that means that particular population, number 25, is in Hardy-Weinberg equilibrium, right? Even though that's not a question, that's something that's interesting. For tasting, there's no issue. No evolution. All right. So we're moving on. Hopefully you get this. You had time in class to do it. I hope you understood it. If you have any questions, please write them down so that we remember them when we get back from spring break. Have a great spring break. See you soon. Bye-bye.